1, 2. Hello, everybody. Sorry, it's been a while. Just doing a quick audio check here to make sure everything is kosher. Make sure everything is kosher. I'm not getting feedback on my own system. The music might be off a bit here. Okay. So using black hole generative music thingy again. So I'm not sure how long I'm going to do this stream, and I don't want to do hours of ambient music because it's quite boring to create, although it's fun to make it initially and to listen to it. But man, it's a chore to sit there and make 4 hours of it, or 2 or 3 hours of it, whatever it's going to be. So, and I can vary it a little bit. There's some cool presets on here I can click while I'm doing the stream, so it jumps around a bit. All right. Let's see what we got here. Just resize a few things here. Okay. I started with a little soil sample here I got today. It's been really hot and dry lately for the most part, a little bit cool at night. So other than going to a pond or the salt marsh nearby, not a whole lot of water samples to get. No pond, no puddles, no standing water of any kind. All the mud is pretty much dried up, so my only choice was to get a dry sample, add a little bit of water to it from a local wetlands by work. It's just dried up right now. They have floodgate control there, so they kind of let it dry up once in a while. So I have some samples of that to look at real quick. That's what you see on the screen here. It's some rehydrated microplant material, pretty much. Let me fix this music volume here a bit. I think that's better. Okay. Let's poke around and see what we can see. I have a lot of plant samples to look at. Thought that'd be kind of fun. I haven't done that for a while. But first of all, let's take a look at this dry turned wet mount. See if there's anything interesting here. Mostly not, I would assume. Just had to give it a shot. Might find some diatoms or something like that. Since they have glass shells, they would persist and have some interesting shapes. That's something interesting there. Some interesting album art for some bizarre album for a band or whatever. All right. Just little bits of plant material. Here's the water already receding a little bit. Nice little grid of cells there. Go a little closer just for fun. more light. So yeah, this is a couple cell layers thick there. Let's fix that brightness a bit here. There we go. A little bit better. Yep, not much to see there. Okay, let's keep looking. There's filaments of stuff. Yeah, this soil is bone dry with a bunch of dried algae kind of draped over the top of it. That's what a lot of these little filaments are. Some nice little cell patches again. Okay, well, it's a cool little shape there. Alright, let's go back to this guy. Alright, so just had to give it a quick shot. Not interesting like I figured it would be. Let's see. Let's take a look at a whole bunch of specimens here. Start with the less hardy ones here. They're starting to wilt. Okay. I'm going to switch to the other scope. So we can do a little more dissecting scope, or otherwise known as a stereo microscope action. It's very, very dim. There we go. Now, let's see what we can find here. So this is... got a couple weeds here to look at since they're so plentiful. And there's a more wide shot there. Alright, so this is wild mustard, related to the one you actually have in your mustard seeds that you have to make your condiment. And it's just the most prevalent weed in the area right now. It's going nuts. Tons of those little yellow flowers. It's actually quite attractive weed, but it grows like crazy, so it just carpets an area even when you don't want it to. But so we can zoom in here a bit on it. Flip it around here. So it grows these little these various stems coming up the side here. This is a growth pattern where, see how the nodes are what they call alternating nodes going up the sides. There's alternate, there's opposite, where they go opposite each other on the stem. And at the top of it, you always have the inflorescence. In this case, it's a cluster. And you can see the venation of the petals there. So we can zoom in a bit on that, you can see a little bit better. Nice venation. Stop moving it because it's too jerky here. Got to fix that color a little bit. Get some better contrast somehow. The fluorescent light on the scope gives it kind of a slightly brighter yellow color than it really is. But yeah, so it's tons of little flowers. You can see that some of the buds haven't opened yet there. And if I could carefully go inside here. Let's see the flower parts. They're the stamens that have the anthers here, which contains the pollen, which, as everyone knows, is plant sperm encased in little packages. And this in the center is the stigma on the top here, where the pollen attaches, and grows a pollen tube to go down through the style, down into the ovary, or in some cases, sunflowers, multiple ovaries at the base of the style. So it's anther on the stamen and stigma, style, ovary on the feminine part of the plant. Now, some plants are male, some flowers are male, some flowers are female. Some are hermaphroditic, like this, where you have both in the same flower. Some have entire plants where it's a male plant or a female plant. In the case of like a ginkgo biloba, which a lot of people take as a extract because it improves circulation in the brain, but it's not really been proven to do much. That's a whole other story, but it's also toxic at a certain amount. You got to be careful with it. But they have male plants and female plants. So for the most part, where ginkgos are grown, because the fruits that they make when they pollinate are really stinky. I've never smelled them, but I've heard it's more like dog crap. And since they drop prodigious amounts of fruits, you would have quite a mess on your hands. So you generally have just the one sex of the tree in a neighborhood, or maybe even in cultivation total. I forgot the whole backstory on it, but you basically just find the one, because if you have a male and female together, you're going to make the nasty fruits, and it's just going to be a horrible cleanup. It's one thing if you have, you know, a nice fruit tree that makes fruits you like, but also makes a mess. But if it makes just kind of stinky fruits you're not going to eat, and it also smells bad, then it's pretty horrible. We have the little clusters here of other unopened flower buds here. So you get a lot of flowers on the head. A whole bunch of flowers. This one's already passed. It's already wilted. So yeah, that's mustard. And they have another common one around here is called wild radish. Looks similar to this, but it's white usually. And it has little fruits that look like a chain radish, not a typical radish that you eat. And I didn't find any of those today, but in some areas they're all over the damn place. So the two most common weeds right now in a lot of areas, you'll find a wild mustard, wild radish, and it's can't miss them. And they are, attractive flowers. It's just because they're weeds. Basically, if you guys don't know what a weed is the generic term weed means plant adapted to disturbed habitats. So if you have construction or a flood or a fire or whatever, and the first things to sprout and spread like crazy, basically what they call pioneer species for an area, but in this case temporary pioneers because they're usually not perennial plants. They are, they're called weeds because they can come in, take over an area when other plants are still trying to germinate or still trying to get a foothold somehow. The soil conditions aren't right, the temperature's not right, the water level's not right, something's wrong. And weeds are very, very hardy. You can pretty much survive. I mean, technically, marijuana is a pretty hardy shrub, but it's also a weed. Technically, it grows in terrible soil in the wild. So a weed doesn't have to be a tiny little smushy thing you can just step on and pick up really easily. It can be very fibrous, it can be more shrubby, but usually weed refers to something that's it's more herbaceous, it's less shrubby, less woody. So some of those plants are kind of in the transition area between those two, depending when you find them. But that's essentially what weed means. Plant adapted to basically thriving in a disturbed habitat. So there are native plants in California that are weeds. I have a couple of videos I put up. Actually, I did some synthetic live streams from some hikes on my other music channel that just happens to have some hiking stuff on it too. And for some of the local parks, and I found one called pineapple weed in a couple parks in Northern California. And it's wonderful. It smells like pineapple, even if it's just a tiny little weed that's you just step on and wouldn't even notice it if you didn't know what you're looking at. But it's very good at you find on trails all the time, which are disturbed habitats, and it grows very, very well in there. So that's technically a weed. So weed does not mean non-native, invasive necessarily in a bad way, but usually because they're so successful, they are invasive. Hence the pejorative for weed. It's not always a bad thing, certainly not for the plant's success, but as far as humans are concerned, they'd be nuisances. Like here you have waterways, you have this tamarisk stuff that got introduced and just chokes the waterways. It's like a large shrubby water weed, or water hyacinth, another one got introduced in some of the waterways, and it grows like crazy. Hey Paul, how's it going? Sorry, I wasn't looking at chat for a while. Got the pineapple there. And it's not Pineapple Express, it's just pineapple weed. So that was mustard. Let's grab a couple other things here real quick. Just go through some quick plant samples just because there's not a whole lot to see here, but something that I could look at other than the water samples I couldn't get today. This is what's left over of wild oats when they have already sprouted and have, let me turn that down a bit, that's way too bright, and actually move this a little bit so it's not quite in the way. Yeah, it's kind of bugging me there, that ring light is kind of being a problem. Anyway, this is the what's left of the essentially like the bracts that are left that hold the fruit for the oat. Once the flower has been pollinated, the fruit forms, and then it drops off. People say, "Why are you calling it a fruit? It's a grass." But flowering plants produce fruits. That's the botanical term, fruit. So it could be a grass, it can be an avocado, it can be an orange, and yes, even a tomato is a fruit because it comes from a flower. It has seeds, and it comes from reproduction. So you can have little tiny dry seed things. You can have grains. You can have fleshy fruits. You can have things that look like something you would never ever want to eat or mess around with, but they're botanically all fruits. Anyway, so that's the little bracts that you get in the oats, and when you see them from a distance, this doesn't go that far back, but you get this long blowing. This is like when you see the fields of grain and blowing golden fields in California and other states. It's tons of these guys left over after they've dropped their fruits, and just because the wind knocks them out, basically. And you're left with these dry husks on the stems. They're quite sturdy too. They last quite a long time. So that's wild oat, also called Avena fatua. And if you ever see that Aveeno facial spread, that comes from the genus Avena. It's Aveeno. It's spelled differently, but because there's oatmeal in the facial treatment, so they took the Latin name and just kind of tweaked it a bit. What else we got here? This is interesting. Speaking of other stuff that's familiar to some people, this is called wild lettuce, and it has lactuca is lettuce, and the leaves are serrated. So the genus and species are Lactuca cereola. And you can see get a better light on this guy. This one has a lot of cool like 3D venation on it too. This is a very sturdy plant, adapted to dry conditions. But you can see the little serrated toothed leaves there. And what's also interesting is on the back side, look at all these spines on the stem. Luckily this one's pretty soft, but the older ones can get quite hard and actually hurt when you try to pick the plant. So if I rotate this a bit, you'll see this spines there. So serrated toothed leaves, little spines on the stems. This is the underside of the plant. I'll flip it on the other side. This is the way you see at the top. So you can see, again, you can see the little toothed serrated leaves. The venation on the top is more flat. But a lot of the business is on the back end here. Those really cool spines. And then the very pronounced 3D venation. You can see it's very rough. It's textured. And when you pick it, it milks. It has a milky sap that comes out of it. There might be some left in here if I pop it right now. I picked a while ago, so it may have already bled out the milky sap, but let's see if any's left here. Yeah. It kind of already came out of it earlier when I picked it. So that's another indicator that that's what you have. They have a milky sap in some of these. Another weed. This is another introduced plant in this case, not a native plant. So most of our plants are introduced from the Europeans coming over here in the old days. And of course, when we're talking old days, we're only talking a few hundred years ago. But so there are tons of things got introduced really quickly in a very short period of time as the settlers came here and brought stuff either intentionally or unintentionally along with their animals or with their cargo or on their clothing. In some cases, some seeds and some stuff. So that's creates a whole other thing. We have other animals brought here too, like the European starling and the house sparrow, and a bunch of other birds and stuff that were brought here that have become kind of a nuisance. Yeah, pumpkins are fruits. Sorry, I'm behind in the chat here. Anything coming from flowers and are pollinated and you know, basically from reproduction that creates a fruiting body of some sort. That's why they call it fruiting body in fungi. It's not a fruit, but it's a process of reproduction. So they call it a fruiting body because it's another form of reproductive output, but it's not technically a fruit in the botanical term. It's a, you know, they have sporangia formed in little structures like a mushroom, or depending on the type of fungus you're talking about. But when it comes to flowering plants, like you'll see some plants say, oh, this is a flowering cherry. Well, of course it is. They all produce flowers. They may not all be viable. They may not all be producing fruits that you can actually eat, or that actually pollinate correctly because they've messed with the breeding so much in them. So they might not create fruits you can eat, but as long as they haven't made them sterile with some kind of weird genetic tweaks or crossbreeding that got some weird results to get a look, they all potentially could form fruits. Hey Gary, how's it going? Thanks for coming. So yeah, that's that one. Let's go to some more plants here. I don't want to linger too much on each plant because I know a lot of people are into botany, but I wanted to have some interesting content that people may not have seen before. This is another weed that grows by the water called curly dock, D-O-C-K, Rumex crispus. And it often has this crazy kind of curly approach, like it's always looks a little bit kind of dry and gnarled and curled, and these pointy leaf tips pointing down. Let's see if I can get it on the camera there. Little pointy tips. This one's starting to wilt because I picked it a couple hours ago. And they get huge, giant strap leaves coming off it, and they change color a lot to reddish and other things. And this is the basically the flower head, which probably has already become fruits. I haven't looked at these under the scope before. Let's have a quick look here and see what we can see. Looks like it's already... these might just be the basic flowers. Some plants have really kind of boring, ugly flowers as far as, like, you know, beauty is concerned. But I think these are just a little fuzzy. See the fuzzy material is kind of mealy looking. They have all these little ways of protecting themselves from drying out, because this was in the hot, full sun today in an area which all dried out oats, like the thing I showed you before. No water to be found anywhere, and this thing is thriving because it's got a tough outer skin, like a cuticle. It's got this woolly, mealy kind of texture on the outside, all these little micro hairs and stuff, to anything to retain moisture. But there's enough airflow and enough exposure to still have photosynthesis and to also do gas exchange with CO2, O2, and water vapor. Anyway, so I've never looked at these before up close. Looks like it could be little fruits, but I think these are the flowers. I have to look some more about dock. I've seen dock a million times in my hand, but never under a scope. But yeah, so that's curly dock. I think I have a bigger piece of it here, just for comparison. Oh, maybe I didn't pick it. Yeah, I didn't pick it. I was just looking at it earlier. They often be quite red. So what happens is the green coming from the chlorophyll subsides as the plant is either going through a seasonal change, or it's starting to die and it's not wasting any more energy on chlorophyll. Chlorophyll is very expensive to make. But so are some of the other pigments, like the red pigments, like anthocyanins, and some of the other like reddish and yellow pigments, and so you'll get these reddish expression of, like, when you get the fall color. That's basically the chlorophyll goes away first, and those other pigments are always there, but the chlorophyll was masking them all out. It's like the dominant pigment. Once the chlorophyll goes away, now you're seeing the red, the yellows, various other shades of orange and things like that. This is a cottonwood leaf. There's a little kind of riparian area in this park I went to, which is no longer much of a riparian area. It's all dried out. But this has the classic kind of teardrop leaf with a wide base. Nice typical kind of soft leaf. This is the underside, so the veins are very pronounced here. And if I do a backlight, you can see, let's see, is this on? There we go. Let's turn this off, and we'll do all backlight. You can really see the venation there, and some of the fine netting of the veins with the backlight going through it. Nice color too. Whoa, blue pumpkins? That's wild. I'm not surprised. There's a lot of crazy stuff out there. I haven't done any cross sections lately, but I used to do it all the time in biology lab as I was getting my degree. But man, it's been years since I did anything serious. I have a microtome. I have a bunch of tools to do it in slightly different ways. But I just, I have to get all my tools together and plan it properly to do that. I haven't even busted out my fancy optics yet on the other microscope. I do the oblique lighting, I do the dark field, I do a bunch of other tricks and stuff, but I have phase contrast optics, and I haven't even put those down the damn scope yet, so I need to, maybe next stream I'll do that. But those are best with living little microspecimens like microbes and protozoa and stuff. So once I have a good water sample next time, I might try that. Anyway, so this is a nice little leaf. Let's change the brightness a little bit on this here. A little less in your face. And you can go to the edge here and you can see some toothing on the leaves. Here it looks very sparse because it's zoomed in, but it has a nice toothed edge to it with little protuberances there. And then you go all the way to the stem here, where it gets, you can see all the veins pop out of the stem there, which is a nice look there. So yeah, and I'll flip it over, you can see the top of the leaf. There's a hole on it there probably from an insect. But yeah, beautiful network. Every time I look at this, I think of the Body Worlds exhibit where they had the human body, particularly the brain, injected with plasticized material, and then they dissolved the tissue, and what was left is the network of blood vessels in the brain and the rest of the body. It's really fascinating. Plants and animals share so much with this kind of plumbing structure. Also reminds me that, and just a fun aside, marine mammals have this really cool adaptation to cold water. Normally you would your extremities would start to lose feeling as you lost your body temperature, and so your core is most important, so you start shunting blood vessels from your extremities to your core, but then you can't move. So once you're unable to move through your environment, you're pretty much toast, especially if you continue to stay cold or get colder. So marine mammals have this very clever adaptation, not only the blubber and the layers of fat they have to insulate them, but they have this really cool structure called Rita mirabile, and it means beautiful net or wonderful net, and it has this network of blood vessels surrounding the blood. So you have the arterial flow going around the venous flow, because the vein always brings the blood back to the heart, and if cold blood hits the heart, you're pretty much done, right? You go into shock, and if your body temperature drops and the heart has sensors in there, it's pretty bad news, as well as other parts of your body getting the cold. So on the extremities, they have kind of like I have insulating cable wrapped around certain types of cables. It's called countercurrent thermoregulation, and they have the warm blood on the outside of the cooler blood vessels cooling, that normally would send cool blood out, and it warms the blood going back to the heart. Really interesting stuff. So I always think of that when I see these little net structures. So that's cottonwood. Here we go. One marsh plant that was out there still, because everything else is dried up and dead mostly. It's one of my favorites. It's called pickleweed, and this is one of my favorites out of the baylands, salt marshes and stuff. And we take the backlighting off, put the front lighting back on, and if you look at it, it looks like a chain of little pickles on these stems. So the top, this little dark area here at the top, is basically, you would have little flowers coming out of those little nodes, and they reproduce just like any other flower, any other plant does. They just have tiny, tiny, tiny little flowers. In fact, there's something going on here. Let's see what's happening here. Other way, other way. I never looked at these up close either. So this may have already gone through some reproduction. I'm gonna have to cut into this some more and take a look at it, but I normally see these little kind of spikes sticking out. Those are like the little flower parts. But this one looks quite different than I've seen it, at least with naked eye. So there's one without the color, just a plain tip of the stem there for contrast. And it's like little plates. It's all like sheathing. And then toward the end here, you got some damage from parasites and other kinds of stuff, it looks like. There you go, there's a stamen right there. Look at that little stamen sticking out of there, little anther sac. So there's the reproductive part of the pickleweed. It's a kind of classic stamen shape, just very, very tiny. Rotate it a little bit carefully. I always have to think in reverse on this stuff, so it's hard to do. There we go. See little notches in it. So it's multiple lobes, just like the other stamen you saw on the other plant. So yeah, there's the reproductive part of the pickleweed, at least the male part. And the end of it. So yeah, some of the animals like this. I've seen a local endangered species called the salt marsh harvest mouse, which is one of the reasons the wetlands like the baylands in Palo Alto and other places are protected. And I've seen them sitting on top of a cluster of pickleweed with basically, you know, a piece about, I don't know, that big or smaller, and eating it like corn on the cob. Sitting on its haunches and had it brought up to its mouth, and it's eating pickleweed like corn on the cob. It's one of the cutest things ever I've seen. And it was special because they're hard to find. As soon as they come out in the open, the larger birds like the great-billed heron and some of the egrets, they go, oh, food, you know? So they're not just going for fish, they'll go for little mammals and other stuff that they can get their hands on. So that was really cute to see that, but he was at risk by coming out to be cute for everybody to look at. So poor little harvest mouse. And then one more fun little thing I got at this location was, I haven't looked at these under a scope either. There's wait, is that the right? Make sure I got the right piece of it there. This is a small flower, actually called lotus. So it's a legume like lupins and other stuff. It has the little banner wing keel petal approach to the flower head, and it's just cute little yellow flowers. and it's I don't know, just really nice to look at. You find them in the lawns a lot, very, very tiny. So I'm zoomed in quite a bit on this guy, but it's lotus. It's kind of fun. Okay, so that's the stuff I got from the park. Now I have some other stuff I had around home that was kind of nice, and right by home, bigger stuff. So this is a fuchsia bush. First I'll start with the leaves. You can see they're velvety and kind of mealy looking. Turn the light up a little bit on this guy. You can see that kind of fuzzy on the edge. Gives it kind of a velveteen look. It's quite soft. I'm gonna zoom in a little bit. You can see the kind of glistening little micro hairs on there. It's very velveteen, and it has that shine you get from velvet, kind of weird gloss. Very nice, soft leaves. And this one's ornamental, some kind of cultivar. So look at the colors on the bottom. Just crazy. Those anthocyanins. Beautiful. This is something Prince would enjoy. I don't think this one transmits light too much. It's pretty thick. Let's see. Oh, there you go. That's really nice. Now you get the hybrid, the purple coming through below, through the green. Beautiful. So there you go, that's this fuchsia leaf. And the stem is that same kind of purplish reddish color. There you go. Looks almost fake, it's so colorful. And then of course the flowers are stunning. Two beautiful flowers that hummingbirds love. It's red and perfectly usable by their long bills. Look how long this flower head is. Oh, the nectaries are all the way down there. And then what I was going to try to do, carefully, is to open one of these up so you can see. Come on. There we go. So on the inside, you can see the white anthers there on top of the stamens. Very mealy looking with all the pollen on there. Right there. So it's kind of flaking off there, as pollen does. You can zoom in a bit on that. Just have to move it over a little bit. Took it out of the tray so it's harder to move. Let's go a little bit more toward the center. There you go. Oop. Stuck on the... There you go. Now I can zoom in. Nice flaky pollen. See the one next to it hasn't really opened yet. It's still pretty solid, but once it kind of opens, the pollen just bursts out like snow. It's crazy. So that's the fuchsia male portion, and I can't remember if these guys have. Let's go look in here at for the stigma. These are all anther. There it is in the middle. It's red. It's camouflaged. Zoom out a bit. It's hard to wrangle that way. And if you look. Back on the tray because it's much easier to wrangle that way. There we go. There's the stigma peeking out and blending in with the flower parts. There's a red stigma at the top there. Right there. And then the style is the tube going all the way down. Down there in the middle. So if I can separate these anthers, or these stamens from the pile. It's very hard to do. Let's see. I need some finer forceps for this, but yeah, you can basically get the idea. So it's in the middle there, all the way down, and if I continue dissecting down, it goes all the way down. In this case, the stamens are attached around the kind of the top of the flower, this little ring. You can see how they're kind of attached here. But the style goes all the way down because the ovaries will be at the bottom of the flower down here. Let me zoom out a bit. Right down there at the base of the flower into the actual. Oh, the flower just came off. To the receptacle. That's the receptacle there. And what often happens in most flowering plants that have big old fleshy fruits is the receptacle often swells, and you're eating the fleshy receptacle of the flower. It develops, it contains the ovaries inside of it, and so that's what you're eating. In the case of something like a strawberry, strawberry not only is not a berry, it's the little dots on the outside of the strawberry are actually the individual fruits. They're called achenes. And so it's those are the individual fruits, and the receptacle that you eat that tastes so wonderful, that's the fleshy part on the outside. That's the swollen receptacle, because remember the ovary develops, the seeds come out of the ovary area for the most part. Some of the plants have slightly different development as the fruit develops, but in the case of a strawberry, all those individual seeds are individual fruits, so they had individual flowers servicing those fruits. That's how tiny everything is. And yeah, it's really crazy stuff. So you're eating the engorged fleshy receptacle of the plant, which is your fruit and the strawberry. It's all kinds of wacky stuff. We talk about fruits of various plants, and the nomenclature is just hilarious. So there's banana's a berry, strawberry's a berry, a cherry is called a drupe. The stone fruit, any of the stone fruits are called drupes. Has a stony pit inside, so like peaches, nectarines, cherries, all that kind of stuff. And in the case of a raspberry, it's an aggregate drupe. Every little fleshy bulb with seed inside is a drupe that clusters together as an aggregate drupe. And then, so you're like, well, technically the banana and the orange have kind of a similar idea with a non-edible, they call exocarp on there. Is a little peel that you peel off the outside of it for protection, and it has it has different names for the different types of berry. So you could have the hold on, I got a Windows Update thing popping up. Don't do that. It's annoying. In the case of the banana, the type of berry is called a pepo. Same with a cucumber. In the case of the cucumber, you can actually eat the peel. It's a much thinner peel. And same with the pumpkin. I cooked it a certain way, you could probably eat the outside of a pumpkin. Although most people don't. They peel it and eat it. That's also a pepo type of berry. In case of a lemon, lime, orange, all that kind of stuff, it's called a hesperidium, and it has a much tougher skin and segments, things that break into segments, unlike the banana, which doesn't really do that, or the cucumber. So, and there's a whole long thing about fruits and crazy names for stuff. Cleavers. Yeah, goosegrass. So goosegrass is fun. The little local one that's small, it's Gallium aparine. I have it on one of my my hikes that I did on my synthetic channel, just because that's where I put some of the hike videos. And goosegrass is funny because that's where the invention of Velcro came from. This French guy who was working with botanical specimens just got the idea of like, wow, this thing is really interesting. Under close inspection, he saw these little hook structures on the fruits, the tiny little fruits of the goosegrass. And kind of like how burr clover has the same kind of thing, these little hooks, but it's much bigger and different shape. And he studied it and he goes, this will make a really interesting fastener. So over a period of time, I don't know all the details. He's the guy who invented Velcro, all based on the way goosegrass clings onto animal hair for transport. So really interesting stuff. We get a lot of ideas from nature. Okay, so that's fuchsia. Fuchsia was kind of nice. Let's look at, here we go, here's jade plant. Never looked at this one either. Here's a nice succulent leaf. In this case, I could probably get a nice cut on this one. See the inside of it. Let's see here. Of course, I misplaced my scalpel, which is great. I have a dissection kit and no scalpel. That is not a good thing. As one of my former biology teachers said, when you did something dumb or bad in class, instead of 10,000 lashes, like with the whip in the old days, you would get 10,000 lashes with a rough endoplasmic reticulum. You know, the packaging apparatus and transport apparatus inside of the eukaryotic cell. It's a super biology nerd joke, but it's pretty funny because you don't use the regular ER. Use the rough ER. It has the ribosomes on it because it's supposed to sting more because it has ribosomes on it. It's just silly stuff. Let's see, I have a small knife here I could use that should do the trick, although it's a bit coarse compared to my scalpel. Let's do a little slice down the middle. It's quite leathery. Do another slice here. Let's give it a little section. Turn it on its side. And you can see this dissected leaf of jade plant. So here's the typical, you know, round, nice kind of smushy leaf with the, look at all that shiny protection on there. It's quite waterproof, kind of waxy, shiny. And so the cut I made is kind of primitive, but over here is the cut turned on its side. So if I zoom in on that. And this one would be hard to microtome because it's very flexible and smushy. But I think with the very careful application of my scalpel when I track it down, I could get a nice thin slice of this for the compound scope. Anyway, so that's the smushy. Look how moist it is inside. There's just tons of moisture in there. But the outside of the plant is totally dry and waxy. This is great protection from the environment. Not just being succulent, but also waxy. You can see those individual go way in here on it. I'm not going to get close enough to see stomata, I don't think, but definitely seeing some interesting scaly structure in the waxy covering. Really interesting. It's almost like the silvery bits in fish scales. Really interesting color there. So that's jade plant. Here's a bigger normal piece of it, uncut on its little stem. Very succulent. And this, you can see it's kind of spoon. Spoon-shaped. Interesting little plant. What else we got? This jade plant. Let's look at some other interesting. Here we go. Let's do some coniferous stuff here. Where's that other piece? Oh man, I must have dropped a piece. Oh, here we go. So I got a piece of juniper here. Should be nice and scaly. I've got juniper, I've got pine, and I believe this is also, yeah, it's another kind of juniper. There we go. Nice and scaly. Look at those scales. Really cool. Again, the ultimate adaptation to dry conditions. Thick, waxy, scaly leaves overlapping, so little pockets of moisture all through the stem there. Let's got another piece of it here. I still have little, and they produce these little berry-like cones, so they're not your typical compound, fancy flowering plants. These are cone makers. Cones are more primitive than your typical flowers. In some cases, they require fire to open, so you don't just drop seeds and waste them on the ground and not be able to germinate. So you burn away the debris, create nice nutrient soil. Also burn away your competition in the area for light and for moisture and all that stuff. And the seeds go right to a nice soil right after a fire. So, some interesting little variations in the shapes here. Looks like some damage that was healed. So that's juniper. And here's another one. Slightly different. Scaly bits here. I couldn't find any cones on these, or little berry-like cones. But interesting little scale structures there. Oh, I didn't know that about the jade plant. Interesting. Yeah, so this is just kind of a fun, alien-looking plant up close. But again, look at that waxy, shiny bits there. It's just perfectly adapted to dry environments without desiccating. That's why they've been around for so damn long. Very successful plants. And then you got pine trees. What I was going to do first let's just take one of the bundles. They call them fascicles. Bundles are fascicles. And it's just make a little sheath, and then you part of how you identify the pine is how many needles in a bundle. So in this case there are 2 on this one. There's the sheath I pulled out of the, let's get it, there we go. There's a little bit of a sheath at the bottom there, kind of whitish. And there are the 2 needles going out. And not sure what kind of detail we'll see here, but give it a shot. We get some color out of it, but not a whole lot of detail on these. There's some, yeah, there's a little bit of ridges there. It's kind of nice. Get a little extra light on here. This is not enough light. I need help from this light as well. Still not that much light. Yeah, Look at those ridges. That's cool. I never saw that before on pine needles. That's wild. I'll have to do some microtome sections of some pine needles at some point. Be interesting to see those. So yeah, that's that guy up close. And here's kind of a overall bushy view of it. Go down to the base here, you'll see the bundles coming out of the stem. Individual sheaths. All kinds of bundles on the pine. And the stem itself is very kind of those scales, flaky scales. Some of the other kind of junk that's kind of accumulated on the scale, on the stems there. So yeah, and they always smell nice too. I love pine trees. So that's some of the conifers there. No cones, just leaves. They also call it gymnosperms, is the generic term, which means naked seed, because when the cone opens up, it's literally just a seed with sometimes a little wing structure on it, so it floats down. If you ever, you can also get them on normal flowering plants too, like a maple tree makes a really cool double-winged fruit that helicopters down when it breaks off. It's always fun to separate them, throw them up in the air. It's like a little, those little called puddle jumpers, little whirly gigs or whatever, spinning as they come down. So they have a nice soft landing on the ground, and then they hit some wet soil, and boom, there they go, start germinating. But the pine, some of the pine seeds, as they come out of a cone, can have a little wing structure as well, so the wind will blow it and it'll disperse, which is pretty cool. What else I got here? I've got a piece of the Dracaena, which is like a type of yucca leaf that was outside. Very strap-like, you know, typical monocot type plant. So there's monocots and dicots based on how they germinate. Either one leaf comes out or two leaves come out, and then the structure of the stem itself is very different. Monocots have certain bundles in their stems, and they can be quite woody. So yucca and agave and various other things like that. But they still can be quite strong, but never quite as strong as a proper dicot with like a nice hardwood. So, but they have like strap-like leaves, kind of grass-like in a way, but very thick. As you can see where I tore it there. It's very moist inside, even though it's super dry on the outside. Again, very well adapted to desert-like climate. Very fibrous, very hard to tear. Even when they're kind of ripped up, they can still hang together. And then, you know, they could heal a little bit and not totally die, but they take enough damage they'll basically dry out. But yeah, these are interesting ornamental plants. Often plenty of, there's several kinds of Dracaenas actually in office buildings and homes that are kind of a softer variety with the curved leaves. Dracaena just means like dragon tree, basically. It's I forgot where the name came from, but it's an interesting group of kind of palm-like plants used often for ornamental use. What else we got here? This was an interesting one here. This one I got the whole weed because it was like, I wanted to get rid of it, but at the same time, like, oh, it'd be good to look at it under the thing. And this is a whole soup to nuts approach. I've got the entire plant from the roots with some soil on it, the leaves going up to the stem all the way to the top. And even the fruits, in this case, It's a it's like a dandelion relative, essentially. I forgot the exact. I think this one is. This is not cat's ear. There's a couple different ones like this. This one's pretty fibrous. But anyway, you get the typical kind of dandelion-like leaves. So this is kind of a wide leaf coming out the end, and some little kind of wimpy scallop parts toward the base of the leaf. Pretty good stem. Lots of flower heads coming off the top. So here's what's left of one of the flowers. And I believe, yeah, the dark bracts. This might be a cat's ear. The cat's ear is usually a little bit wimpier than this. Anyway, it's an invasive weed, kind of like a dandelion. Nice, colorful yellow flowers that open up and eventually produce these little pappus, parachute-like structures that once this thing opens up, they catch wind and float around like little parachutes. It's interesting. Look at that, how that's kind of torqued around, almost like an alien hand or something like that. It's a really bizarre structure there. But yeah, as the fruits are developing inside, and little tassely called pappus structures come out, eventually it will dry out, it will open up, and the wind will blow these away, just like on a dandelion. It's just a much bigger plant and different species than a dandelion. So yeah, that's one of these little guys. And of course the plant is too big to put under the scope, but I can actually take some of the roots here. See what this looks like under the scope. So I'm cutting this. It's pretty fibrous at the base here. Yeah, that's not going to work. I'm going to break off some of the smaller roots at the bottom. Let's see here. Let's put a little protection down so I don't cut the wrong thing. There we go. Put the whole chunk with the soil and all on my little stem there, on my tray. And there's the root structure of this plant. It's got the classic wandering root pattern, much like you get in fractal designs. Like, have you ever seen a 3D simulation called L systems, where you actually mimic plant root growth and some of the other plant stem growth, and you get these wild, kind of torquey, almost Tim Burton-like tendril thingies in the root structure. So these are the main root. Basically these kind of plants have a taproot, and then these little kind of accessory roots that come off. So the taproot is what I'm holding right here, and then it branches out for kind of peripheral roots that go out for stability and for nutrients. And looks like this one actually got around a screw. Wow, that's bizarre. There was a screw in the yard, and this kind of grew around a screw. That's funny. So that's that dirt clod there. There's a screw in it. But you can see the individual like rootlets, and if you go super close, under proper magnification, you can see the root hairs, the actual fine structures that actually involve in the water and nutrient exchange. But way too small to see in this kind of setup. But it's kind of fun to see like the plant broken down to all the individual parts. So I have the entire plant kind of dissected down. So yeah, there you go. That's that one. What else do I have? This olive tree leaf. Kind of boring, but it was something else to grab. Again, very mealy, kind of hairy underneath. Definitely too thick to distribute. Yeah, so you can't get too much light transmission through it. It's very thick. I'm gonna if I go back to the top light, and you can see how it's kind of curved over at the edges. Again, helps create a little shade for the underside of the leaf, a little microclimate, so it's not so hot and not so dry. And so the thickness of the leaf, the hair, the curvature. Here's the top part. You can see how it's curved, very, very curved. Also very shiny, waxy, to retain water. Nice green color there. And so yeah, that's the olive tree leaf. That's kind of all I got for the plants. What else do I have to look at? Oh yeah, some wonderful, well, kind of wonderful, kind of nasty stuff from the fridge. Stuff goes bad, you have a microscope, you look it under the microscope. So here's some stuff that I kept in the garage for a while. I wanted to desiccate. Yeah, the soil here is pretty rough. Northern California has very, very kind of heavy clay, rocky soils. I know, because growing up in a different city near here as a kid, many, many years, doing all the yard work and digging things up and stuff, I was digging tons of rocks out of the soil all the time. You had to basically soak the ground every time you wanted to dig anything. Clay is just brutal here. And also it takes a long time for the water to seep down because of the fine particles of clay. So you have to basically, when you move into a house that hasn't had a lot of work done, a yard or whatever, you have a lot of amendment to the soil to do. A lot of either rototiller, a lot of handwork, lots of, mixing in various things to make the soil even usable for growing something, or to make a better lawn, or just easier to in the future dig and put post holes in, or whatever you want to do with your yard. So yeah, it's high clay, rocky soil, very crazy. What else we got here? So anyway, in the fridge I had the remainder of some refried beans that were natural. They weren't nasty processed beans. They're pretty good quality. And I left a little bit of it in the fridge too long. So I'm like, oh man, it's fungus city. So I put it in the garage for about a month, let it totally desiccate. And so I'm gonna put a little bit of that on the, well, first on the dissecting scope, just to see what we gotta see. And then I'll make a quick slide of it for the other scope. So here's a fragment of it for browsing in low power. Just looks like gray fuzzy dirt. All those spores, just solid fungal spores. Looks like dirt, but it's almost solid fungal hyphae and spores. Very mealy, dusty looking. Look at all that dust. Spores, spores, spores. And it started out as green, of course. That's the most common spore in a lot of these fungi that grow on your food products in the fridge and stuff. But now it's all gray. Big slab of gray spores. So what I'm going to do real quick is I'm going to take a little bit of this debris. I'm going to transfer it to a cover slip and then do a dry mount. That first, dry mount on the other scope. So now I'm going to switch cameras again. This guy up. And now you can see all the hyphae. Look at all the dried up hyphae from the fungus. just totally took over that. What was left of those beans on the top. And this is just the lightest touch to the dust attracted to the cover slip. So I can zoom in a little bit here. Some nice dried up hyphae. Looks like those little roots. There's the same twisty turny curvature like those little roots did. So yeah, tons of fungal hyphae all clustered and dried up. See if I can do a quick dark field, get a little punch on here. It's not really cooperating with the dark field. Come on. You can do it. Apparently you can't do it. Let's try this one instead. Not that one. Not that one. That one might be good. Nope, it doesn't work well either. Try this one. Oh man, it's not cooperating. Let's try one more. That one might do it there. Getting bits of it. It's not quite what I wanted, but it just gives you a different textural view of the structures there. So now you can see some of the finer details that didn't show up in the normal bright field. Oh, thanks Gary. Thanks for dropping by. I got have more water samples next time. It was just a bust out that everywhere I went was all dry, so catch you next time. Alright, let's get a close up on these fungi. That was the dry mount. Let's do a wet mount now. I'm going to take another slide, another cover slip. This time I'm going to put a little drop of water. Slide, and then I'm going to do the dry transfer again from the fungal mess to the water. Let's see what kind of magic we get. Might be a little more interesting this time. So the water created little pockets of spores, looks like. A lot of air trapped around those little microspores. So there's a lot of it's hard for the water to wet it once it's dry. So you get this kind of these weird little fragments of things that you need, like a wetting agent, like a surfactant or something like that, to basically make the spores get wet easily. makes some cool little shapes though. Almost like nerve cells. It's also perfect for Halloween. It looks very Tim Burton-y there. Lots of swirls and curls and funny shapes. Alright, I'm gonna pick a spot and just zoom in and try to see if I can find individual spores clearly. Let's try this one, it's more fragmented. There we go. Now we're getting individual spores here. Let me give some more light. And you can see the little spheres there now. Those are the spores. Just need a little more power. They're pretty small. And just to make it easier, and I'm lazy, I'm going to use the software zoom and not the microscope zoom. So you can see the spores better. Nice little spheres. Let's get a bit better contrast on this. Get that hand out of there. Focus, and there you go, fungal spores. So as you can see how that clustered, like I was guessing, when they touched down in the water, the coating on the spores seemed like it had the protection from being wetted, so to speak. So the water more cluster them together in clumps versus having them kind of sit and hydrate properly, like something that's already wet would be in the water environment. So you get these weird little clusters like that. It's kind of interesting how that worked. And you have a few individual spores here and there that are loose, and other ones are big, massive clusters of spores, so much so that it's dark. Islands of spores, and then some individual spores. So yeah. And you can see I'm going quite high now on the power. This is 400 power. And look how many spores in a very tiny area of 400 power. And because they're so light and airy and carry so easily in the air for days and weeks and months, so much so they're in the upper atmosphere, they're in every room in your house, even clean rooms have them, they're just filtered out more than anywhere else. But this is why you can see people have respiratory problems. Because fungal spores are just, I know that people have pollen issues, but in the case of fungal spores, you can get aspergillosis from the aspergillus spores. That's supposedly what killed the Egyptian tomb robbers and early explorers that cracked open the old tombs, and aspergillus spores survived, for basically 2,000 years in some cases. And once they got in a nice wet environment of somebody's lungs or nasal cavity, quickly caused severe illness and death, which they call, oh, that's the curse, that's the mummy's curse. It's like, nope, it's aspergillosis. So, but this tiny little sample had this much stuff in it, and I'm on 400 power, and you can't even count how many are here. If you're able to separate the clusters, you probably easily got a couple thousand in here, and that's the tiniest little sample of this collection. And the dusty sample. This is why I get very careful with these fungal spores. I kept in the garage, I waited until they dry out. I haven't opened it till now. I've been very careful how I'm handling it, and I'm not disturbing it too much to breathe in a bunch, have a bunch get in my lungs. This is not aspergillus, but other fungal spores can cause severe health problems for people, especially if you're allergic on top of that. So as much fun as this is, you do have to be careful. You don't want to breathe in a bunch of extra fungal spores than you already have to, because like I said, the environment creates so many They can do air sampling way up in the atmosphere with special planes that go up there and collect air samples. And they got the pollen, fungal spores, all kinds of other stuff floating around up there. Easily carried by the wind. So there you go. That's what I had in my fridge many months ago. And let it dry out for your viewing pleasure. So again, if I go out, you won't be able to resolve the individual spores so much. You see them here as, well, you can see a couple, if I use the pointer here, there are a couple together there, maybe 2 or 3 together. Or 3 or 4 or 5, something like that. But it's really hard to see individual little, that might be one individual right there. But they're quite small to resolve at lower power than 400 power. So that gives you an idea how small they are and why they carry so well. And just get into everything. You know, you just touch it and you brush it on your shirt, and now you've carried it somewhere else. And then you change environments, and the wind blows your shirt, and a blast of air blows the spores some other part of the room, and then it's just like one of those Rube Goldberg kind of things where it's just a continues going on and on and on, spreading from person to animal to equipment to whatever, you know. Explains why this stuff is really hard to get rid of when you have a problem. So there you go, that's spores. What else do I have? It's kind of it, really. I don't want to make too long of a show tonight, and I'm sure a lot of you guys have already checked out or soon going to bed, whoever's watching this. Are you still there, Paul? Hey Matt, how's it going? So you're UK, right? So early morning for you, must be 7 in the morning, something like that. Oh, cool. You're still here, Paul. Nice. Do a quick review for Matt since you showed up. I was doing a bunch of plants tonight because everywhere I went today after work was dry. No puddles, no nice ponds nearby to easily grab a sample. The traffic was bad. I didn't want to go to the salt marsh and get a sample. I'll do that probably over the weekend. I have a new, I've actually bought some equipment for sampling purposes. Some of these places are really cool to capture specimens, especially the salt marshes, but the trails and the levees are so far from the actual water, and they don't want you climbing down there for good reason. You're disturbing the habitats. Also, you'll lose a shoe. There's so much soft muck, very fine mud, that it creates a suction. If you step down in there, it'll pull your shoe right off and you'll never get your shoe back. Plus, you're damaging the environment. It looks like somebody messed it up when you walk through there and crush some plants, and I just don't want to do that. I want to just collect some nice water samples and leave. So I bought one of these really nice painter poles, which not only goes 7 feet out of the box, but if you're crazy enough, you can extend it to 25 feet. So in almost any condition now, I can put a little sample bottle on the end of it. It's basically a painter pole with a spray attachment thingy for like a paint sprayer for remote paint spraying, or a roller attachment, and then you put a sample bottle in there, and I can grab water samples from anywhere now without having to get muddy or having to destroy the environment or all that kind of stuff. So I'm really excited to start using this guy to get samples all over the place. So yeah, it looks like a crazy hiking pole because it's like 7 feet tall just to start, but it has 3 segments that come out of it. So you can go up to basically 20, I think it's like 25 feet, which is totally insane. But it's really sturdy, so I tested it, and you can go crazy with it, especially if you don't put a lot of weight on the end of it. So going forward, I'll get much more water samples where I want to, when I want to, without having to worry about, you know, things drying up, being too far from the trail, or getting in trouble for going off trail in a reserve or whatever. So yeah, no water samples tonight really, but So I got these plant samples if you go back and watch the stream. But this was, we'll zoom in on here. You can see these little clusters of junk. And what I did is some kind of old refried beans I had in the fridge for too long started to get moldy. I mean, oh great. So I put them out in the garage, let the mold get further worse, you know, much more growth, and it's covered with plastic wrap so it can't go further, and it creates a perfect environment for the fungus to go nuts. It's nice and moist, it stays moist until they populate and use up all the water, and then it desiccates naturally. So I left it in the garage for about 2 months, and I thought this will be good for a nice little demonstration of fungal spores from your fridge. And then I did a dry transfer using a cover slip, and that was the first slide. This is the wet transfer, where I basically put the dry cover slip inside a drop of water, and they naturally cluster together because of the kind of the textures on the outside of the spores don't allow wetting. You know, they're kind of like the way they're packaged on the outside. They're so dry. They need to be kind of in the water. I bet if I let this for a while, they would slowly separate. But I just made this so they cluster like this, and I have to go all the way up to 400 power to actually resolve individual spores nicely. And these are, I'm gonna focus the wrong way. They're the individual spores at 400 power. Nice little spheres. And you can see how most of them cluster together to make these kind of black voids. But that's them individually. And at 400 power, you're seeing like thousands of them here. So it gives you an idea how many were on my food specimen in the garage. And if you sneeze when you had it open, or wind blew, and all that blew around the room, and you're inhaling that. And if you're allergic or just happen to get too much of it in your system, you can get really sick potentially. This is the curse of the pharaohs, the King Tut's tomb, and all those things. This 1,000 plus 2,000-year-old aspergillus spores. These guys inhaled, not knowing anything about it, and died from horrible lung infections. So you want to be very careful with this stuff, whether you're just throwing it out or doing specimens like I'm doing to look at it, and not stir it up too much, breathe it in, because you can't get away from fungal spores or pollen. It's everywhere in the atmosphere, in every room in your house. You know, it's up in the upper atmosphere. You can't get away from it. But it is fascinating to look at it and to think about how many spores are out there, and how many pollen grains are out there. In the case of spores, it's just you know, a little packaged little body ready to create a new stem of hyphae, and then it just binary fission spreads. So you can have one fungal hypha divide into this ever branching stem, and it can take over several acres of land from one hypha. It's like a world record. One of these ones, I think it was several acres at least, maybe more than that. There's actually a couple dozen acres, I think. So this stuff is very powerful. This tiny little spore can, over time in the right conditions, grow to be this massive organism, way bigger than a blue whale, which is the biggest known animal, even bigger than the dinosaurs. And yeah, it's pretty crazy. And then of course pollen is, you know, a reproductive part, but it by itself doesn't do anything. It needs to come in contact with the right species of flower to pollinate. Just like humans, you have to have a sperm going to an egg. Well, pollen is essentially a little packaged sperm. It has to touch the right part of the plant, forms a pollen tube, sperm swims down there and pollinates the egg. So you're basically inhaling sperm. It inhales the pollen, which is pretty funny. Yes, basically flowers are busting all over the place. It's just a giant porn movie. Nature's a giant porn movie. So yeah, it's a funny way to look at it. Yeah, mushrooms in your bedroom, you got to be careful with that, because again, once they Mushrooms are the fruiting body of the hyphae. So the mycelium is the entire structure of net-like. I have some over here, I think on this slide somewhere. Let's see, do I have any hyphae on this one, or did I transfer enough of the spores? There's a nice chain of spores there. There should be a couple hyphae in here somewhere. My original slide was a dry transfer, had a lot of hyphae fragments on it. This is pretty much all spores here. That's not normal hyphae there. It's a vegetative piece of something. Just trying to find a nice filament here. God, look at all the spores. They're just all clustered together. So many of them. Yeah, my other slide had the hyphae on it, pretty much. This is pretty much all spore transfer. Anyway, so the hyphae are the threads. They grow together to form a mycelial mat, essentially. Just a big, to the naked eye, just a big fuzzy patch. Like you know, a bunch of hair piled up. And that grows into the big structure. And then from that, you get the fruiting bodies at the right time of the year, the right conditions, called mushrooms, or asci, depending on the type of fungus you have, or various other kinds of structures you can get that aren't mushrooms, puffballs, things like that. And once you get the mushroom, the gills, or if it's a pore-like one, like a bolete or something like that, they're producing spores, whether you like it or not. And if you grow those in your room and you have some kind of ventilation, it's going to blow damn spores all over your room. You're inhaling tons and tons of spores, which is generally not a good idea. So I'm sure the person growing them knew that, but in case they didn't, you may want to tell them, because you don't want more spores in your body than you're already inhaling on a daily basis, especially if your health is poor. I mean, if somebody was more prone to COVID and more prone to other kinds of lung infections and other kinds of things, they definitely don't want to be inhaling spores on a regular basis more than what's in nature. So just one of those things to think about. A lot of people do this stuff and later on wonder why they got sick, or wonder why they feel weird or whatever, and they expose themselves to more than they should have. Just something to think about. Anyway, that's kind of, I think I'll go and do it. I've already gone for at least 2 hours, I think. Normally I do a 3-4 hour show, but I ran out of specimens. I didn't have a bunch of water samples tonight. So I finished off with my dried up fridge sample, fungal spores. I'm going to get some nice water samples, hopefully this weekend. I have some time. I got to work on a hardware jam. I got to work on some other stuff. So I'll squeeze it in somewhere, along with some sleep. But I took a break for a while just because too much other stuff going on, but I'm going to start doing this more on a regular basis. So just keep your eyes peeled. I don't have a structure yet, a schedule, but I'm definitely going to try and do at least another one this weekend if I can get some samples. Maybe tomorrow afternoon, get some samples somewhere, or maybe even Sunday. We'll see what happens. But once I get some really nice water samples, I'll be going crazy with the streams again, because that's my favorite. That's where you see the most activity and weird stuff and all that. So thanks for tuning in, everybody, and feel free to always ask questions on here, or if you watch the stream back later and some parts you missed or want to review, put questions in the comments, whatever, or requests for stuff. If you want to see certain things, if I can oblige, I will, because I love all this stuff, looking at fruits and plants and dust samples. I tried getting some dust. Apparently the carpet here is in good shape, because I took several different dust bunnies and other kinds of stuff. I had no dust mites, so that was a nice affirmation that this place is pretty clean. But I haven't done Demodex face mites yet, so I might do that. You guys probably heard about that. I talked about it on one of Dom's streams, the Mr. Wiggly streams, many years ago, about a year ago probably. And it was a great fun to gross everybody out because he looked up the video of the Demodex face mites. And they're on your face whether you like it or not. They live in our dead skin cells. They love it. They just clean up the mess. They don't hurt us. And they're never going away. You can wash and scrub, and they're in your pores. They're on your body whether you like it or not. And they're just kind of funny little critters. Easy to capture with some scotch tape on your head and a transfer process. So I might try those on a stream one time just so you guys can see that. Although it might gross you out a bit, but it's nature, it's your body, and you got to deal with it because it's not going away. And hey, it's free specimens. I just have to get it off my head, off my forehead. So good stuff. So yeah, that's it for now. Next time I go for a hike too, I'm gonna get some more tardigrades. I love the tardigrades. You get them in the water. You can also get them growing on moss. They often, when the moss dries up, you can rehydrate them and they come out of stasis. And they're just one of the cutest little things ever. Little tardigrades. So I might grab some of those too pretty soon. So anyway, I'm babbling at this point. So thanks again, everybody, and stay curious. Again, ask me any questions you want in the comments. And I'm sorry I can't give a direct, an exact schedule of what I'm going to do this next because it's kind of random. But keep your eyes peeled. And I usually do them for at least a couple hours, sometimes up to 4 hours if I'm feeling up to it. So generally that's enough time for whatever time zone you're in. Most likely you'll be able to catch a piece of it live if you want. I try to do it in the evenings. That way UK has a chance to get it if they're morning people. US has a chance to get it if they're night people. And somewhere in between. So seems to work out that way. But anyway, thanks again, everybody, and I'll see you soon.